Today is on the 11th June 2020, so it's my pleasure to welcome Associate Professor Ivan Poon. He is from Trobe Institute for Molecular Science, Trobe University in Australia. So Ivan completed his PhD at the John Curtin School of Medical Research at Australian National University in Canberra. He joined Trobe University in 2009 as postdoc fellow. And since then, he visited Center for Cell Clearance at University of Virginia as assistant professor, and then returned to Australia to Trobe Institute for Molecular Science in 2014 to establish his independent laboratory. The focus of his team is to define how and why dying cells disassemble into smaller fragments called apoptotic bodies. So without further ado, let's welcome Ivan to give a talk on the last dance of a dying cells.
Oh, good. I'll start. So thank you again for the invitation to speak today, to share some of our work. It'll be great to get some input from the EV field. So the focus of my lab is just trying to understand how dying cells break apart, in particular how apoptotic cells break apart into smaller fragments called apoptotic bodies, and how and basically why we actually need this process. I call this talk the last dance of a dying cells. It's not trying to copy off the Netflix documentary from Michael Jordan, but it's that when cells actually die, they actually can undergo some quite remarkable morphological changes here. So I'm just going to show you this video of a monocyte THP1 cells induced to undergo apoptosis by UV irradiation. The cells will first round off, and eventually what you can see is they will form bleb on the surface and then protrusion formation and pushing the blebs away. And sometimes you can also see the protrusion would become beaded and then undergo fragmentation. And so this is the process that we study in terms of generating apoptotic vesicles. I'll come back to this a bit later. So there's four main focus of my lab. One is to develop better methodologies to study how cells actually break apart in the formation of apoptotic bodies. I guess the major focus is at this stage is the mechanism of how this occurs and also the function of this process. We also have a drug discovery aspect as well. But the focus of the talk today is more on the mechanism and the function of this process. So I'm very lucky to have a fantastic group of students and postdoc working on this area with me at La Trobe. And most of the work that I'll present today is done by two very talented graduate students, which is now postdoc now, Rochelle Teixeira and also Georgia Atkins-Smith. So we kind of came into the EV field from a different area called apoptotic cell clearance. So basically, billions of cells will die every day in the human body. And it's very essential that those apoptotic cells are actually being cleared efficiently, because if they're not cleared, the cells can undergo cell lysis and release cellular contents that could actually promote unwanted inflammation and so on. So the apoptotic cells actually have some very clever ways of actually, aiding the rapid clearance of themselves. One of them is the ability for them to release chemotactic factors so they can recruit phagocyte towards where the dying cells is. And for example, there can be soluble factors such as nucleotide, and they can also release smaller vesicles for recruitment as well. It's known for a very long time that when cells undergo apoptosis, they'll undergo blebbing, just circular bulges, and eventually they'll form this smaller fragment called apoptotic bodies. And then the apoptotic bodies or apoptotic cells can expose certain molecular factors, for example, phosphorylserine, which can then be recognized by phagocyte and facilitate the clearance. But what's interesting to us about 6 to 7 years ago is that how dying cells actually then break apart into this smaller fragment is actually not really well understood. So for a very long time, people think that when cells undergo apoptosis, they can generate membrane blebbing, as I mentioned before, the circular bulges on the cells. And people thought that this will be the only process necessary for the cells then to generate the smaller fragment called apoptotic bodies. But we discovered that there's another step in this process. Basically, after cells has undergone membrane blebbing, the cells are able to form various protrusions to aid the formation of apoptotic bodies. In this case, we call them apoptopodia, which are called feet of death. So basically, the protrusion, very thin protrusion, basically help to push the blebs apart and then undergo fragmentation to generate apoptotic bodies. And then this apoptopodia can come in two different flavors. One of them is more string-like protrusion. We call them apoptopodia, and it can be found in T cells, fibroblasts, or endothelial cells, where the function is basically to push the blebs apart and eventually fragment to form apoptotic bodies. But certain cell types, in particular monocyte, that we'll go into a bit more later, is that they can form this protrusion, that is apoptopodia, but then the protrusion become beaded and eventually undergo fragmentation to generate apoptotic bodies. So those are two different protrusions that we have discovered that can aid the formation of apoptotic bodies. So here's just another video showing a THP1 cells undergoing apoptosis, then forming some protrusion again, and then pushing the blebs apart, and then breaking apart. So based on these findings, we propose that how cells break apart is regulated by three distinct steps. Step 1 is the ability to form membrane blebs. Step 2 is the formation of this apoptopodia to separate the blebs or the protrusion themselves. And then fragmentation of that can lead to the formation of apoptotic bodies. And obviously, we're quite interested in the molecular factors that actually control each of those steps. So firstly, I'll talk about step 1. So it's known for a very long time that during apoptosis, caspase can activate a number of different kinases, such as ROCK1, PAK2, and LIMK1. And that will lead to the activation, which then drive ectomyosin contraction to lead to membrane blebbing, and subsequently the formation of apoptotic bodies. However, previously we don't know, which one of them are the key regulator of this process and whether they have actually some sort of redundant function. So to address this, we initially perform a inhibitor studies. So basically we use T cells, monocytes, and epithelial cells as a cell model in this study. And then we treat cells with, treat cells with the various inhibitor targeting those different kinases. And then, for example, here, when we induce Jurkat T cells to undergo apoptosis, it's only when we put the ROCK1 inhibitor, we can actually block membrane blebbing and the formation of apoptotic bodies. We also target each of those genes using a knockout approach. It's only again when we knock out ROCK1, then we can prevent the cells to undergo membrane blebbing and also formation of apoptotic bodies. So this study, we argue that for certain cell types, ROCK1 is key in driving the formation of membrane blebs and the formation of apoptotic bodies. So next, we try to identify what are the molecular factors that regulate the formation of the protrusion that I mentioned before, apoptopodia, that drive the disassembly process. And we took three different approaches. So first is we performed drug screen with the Ravichandran lab at University of Virginia. We also did proteomic analysis with Suresh Methvinen's lab at LIMS, and also a genome-wide CRISPR screen with Marco Herod at WeHi. But today I'll just talk about the first two here. So in terms of the drug screen, it's actually quite simple. So we've got some human Jurkat T cells where we can induce them to undergo apoptosis by antifas, which then most of the cells will actually undergo membrane blebbing. But what's interesting is that only about 10 to 15% of them will actually form the fragment of apoptotic bodies. And then what we did was we developed a very robust flow cytometry assay where we can actually accurately quantify the ratio of apoptotic bodies compared to cells that are actually undergoing apoptosis. And then we perform a drug screen using a library of drugs called the LOPAC, which you can just buy off the shelf from Sigma. It contains 1,280 drugs that targets a variety of different processes. So therefore, and a lot of those drugs have no molecular target. So what we're trying to identify is that if a particular drug can actually either enhance or inhibit the cell disassembly process, we're actually working out what is molecular factors regulating this. When I first proposed this, I think even my own lab thought that was a bit crazy, but it actually worked out quite well. So in the screen, we actually identify a number of drugs that can actually help cells to actually break apart more than usual. And one of them that I'll talk about a bit more is this drug called tropofoxin, which is an antibiotic. We actually identified quite a lot of drugs that can actually block the disassembly process, which is a bit concerning because 1 in 10 drugs can actually block disassembly. But we did use 10 micromolar to do the screen. So we are still trying to work out whether some of those are actually specific or have other off-target effect. But for the purpose of today, I'll just talk about most with the enhancement part. So trophloxacin is a fluoroquinone-based antibiotic, and it kills bacteria that targeting the topoisomerase. It was actually withdrawn from the market due to serious side effect. So therefore, this drug is somehow targeting some human protein. And in our assay, in a model where we induce Jigger T cells to undergo apoptosis, if we do this in the presence of trophloxacin, we can actually promote the formation of apoptotic bodies in a concentration-dependent manner, whereas the two closely related antibiotics can't actually do that, suggesting that trophloxacin is actually hitting something in mammalian cells, in particular affecting the cell death process. So then the next part is going to be pretty tricky. We're trying to work out what exactly this antibiotic is actually targeting in mammalian cells. But we were extremely lucky because in our screen, we actually used this dye called topo3. And this to see how this dye actually uptakes into apoptotic cells. And we find that this antibiotic, trophloxacin, can actually block the uptake of topo3 into apoptotic cells. And how the topo3 actually enters apoptotic cells is through a channel called pannexin 1. So during apoptosis, this channel called pannexin 1 is cleaved by caspase 3 and 7 at the C terminus, and it allows the release of ATP and other metabolite from the apoptotic cells. And the function of that is to either recruit phagocyte or kind of establish an anti-inflammatory environment. But what's very convenient is that this channel also selectively allows the uptake of topo3. And we find that trophloxacin is actually blocking this channel by showing that it can actually block the uptake of topo3. We also show that it can actually block the uptake of ATP, release of ATP, sorry. And then with quite a few other assays, we show that this antibiotic can actually directly inhibit this pannexin channel. So putting this all together, so when we induce cells to undergo apoptosis, the cells undergo blebbing, but if they also occur in the presence of this antibiotic, trophloxacin, the cells are more likely to break apart. And we find that trophloxacin is actually blocking the channel pannexin 1. And we also confirmed that pannexin 1 is actually a negative regulator of how cells break apart. So either blocking by drugs or we express the dominant negative mutant to block the pannexin function, cells are more likely to break apart. But what's very interesting is that when cells break apart, we find that they form this very thin membrane protrusion that I mentioned before, apoptopodia. So based on these findings, we argue that pannexin 1 is actually a negative regulator of apoptopodia. So when we lost the pannexin 1 function under those circumstances, cells that form the protrusion after membrane blebbing to help separate the blebs, and eventually the cells would then break apart. That's the key negative regulator, pannexin 1. But we also took a different approach with the beaded apoptopodia. So this is the protrusion that I mentioned before that's formed specifically by THP1 monocytes. So a few years ago, we developed a really basic method that we can enrich the beta apoptopodia. And the idea is that we're trying to work out what kind of proteins are enriched in there. And hopefully we'll find out potentially what drives the formation of the protrusion and maybe what's the function of some of those vesicles that are released. And when we did proteomic analysis, we found that one of the protein called plexin B2 is actually enriched in the protrusion. And we got interested in these proteins because it's known to regulate cytoskeletal changes. It plays a role in axonal guidance or macrophage migration. And we thought that that may be playing a role in the formation of protrusion as well. When we look a bit deeper, we found that we work out that plexin B2 is actually being cleaved by caspase, although how this regulates function we still don't know. But what's interesting is that when we knock out plexin B2, initially we were quite disappointed because the cells were still forming the protrusion. But what's interesting is that the protrusion are less likely to become beaded, because if they're not beaded and they actually retract much earlier, they're less likely to fragment. So meaning that plexin B2 is a positive regulator of this process. So putting this together, we've identified pannexin 1 as a key regulator of negative regulator of cell disassembly by regulating the formation of apoptopodia. We also find that plexin B2 is a regulator of beta apoptopodia, but it's probably more limited to certain cell types that actually do form that type of protrusion. But after knowing, identify these molecular regulators, now we can actually build some new tools to actually try to understand the function, or further understand the disassembly process. So in collaboration with Michael Olsen from the Business Institute, he was able to share with us the ROC1 caspase resistant mice. So basically, as I mentioned before, ROC1 is cleaved by caspase 3 during apoptosis, and they get activated to drive the blebbing process. And in these mice, the caspase cleavage site is mutated, so therefore caspase can't cleave it anymore. Hence, ROC1 cannot be activated during apoptosis. And we hope to find that, if that happens, we may actually be able to block the formation of apoptotic bodies in vivo. And to complement that, we actually generated the pannexin-1 caspase resistant mice. So again, as I mentioned before, pannexin-1 is activated by caspase. And in this mice, we mutated the caspase cleavage site. So therefore, it won't be activated to become a negative regulator. And we hope that in this mice, cells are more likely to generate apoptotic bodies. And we'll hopefully, validate that they actually do have a change in formation of apoptotic bodies in different settings, and trying to understand where they, have, certain, disease as well. Obviously, a lot of the work that we have done so far, to identify, the protrusion and the blebbing is done under in vitro conditions, but we really want to see, what happens is in vivo. So recently, we've also embarked on looking at this in using the zebrafish system. Obviously, this is quite new to us. I'm not a fish expert. So luckily, we have got Sarah Cassinis from the University of Virginia, and also Christina Kitely from the Trobe, to help us to actually establish this. So we're still at very early stage in this process, but at least, what we did was we got the fish line whereby the cells will excrete, annexin 5, and this protein would then bind to dead cells, in particular apoptotic cells, and then we're able to see some of the cells that are dying were able to form protrusion that comparable to maybe what we see in vitro. And then also, we're trying to capture this process of, the ability of cells to form membrane blebbing, and then eventually, becoming annexin 5 positive here, and then forming apoptotic vesicles. And we're trying to see, whether we could potentially see protrusion between those blebs as well, to see whether apoptopodia do form under in vivo condition. And also, since the zebrafish have the homolog for ROC1 and pannexin-1, we're trying to evaluate, whether they do play a role in the fish system as well. So that's pretty much the mechanistic stuff that I want to share with you guys. The next part is to try, we try and understand, why do we actually need cells to break apart? And we look at this in 2 different scenarios. So one is to look at this in the area of cell clearance, because we think that was been hypothesized for a very long time, that when cells do break apart, it's a way to aid cells being removed by the phagocytic system. Obviously, we're also trying to see whether those fragments of the dying cells do play a role in intercellular communication as well. So to address the cell clearance problem, just going back here with the human Jurkat T cells. So as I mentioned before, ROC1 and pannexin-1 are the key positive and negative regulator of this process. So when we knock out these genes, we were able to create these 3 different scenarios. So when we knock out ROC1, the cells don't bleb, and they actually don't break apart to form apoptotic bodies. Our control cells, as I mentioned before, about 10 to 15% of them will do still break apart. Whereas the pannexin knockout cells, pretty much every cell would break apart. And then we feed this to the phagocyte and see whether that will change, whether improve their phagocytic activity. And when we feed this initially to macrophage, they seem to have no problem actually engulfing cells that didn't actually break apart. But if cells actually do break apart more, this seems to like to, more cells will be able to engulf the apoptotic vesicles. But when we do feed this to those 3 different cells undergoing apoptosis to monocyte-derived DCs or fibroblasts, they seem to struggle with eating those that didn't break apart, supporting the idea that cells fragmentation can aid the cell clearance process. We can also create a different scenario using the THP1 monocyte that I mentioned before, where plexin B2 is key, well, is important for the formation of the beta protrusion and then undergo fragmentation. And when we knock out plexin B2, the cells can still form protrusion, but the protrusion don't form less beaded, they will retract quicker, so therefore they will not be able to undergo fragmentation. And again, when we feed this to phagocytes, in this case, epithelial cells, if the cells are not able to break apart, they're less likely to be engulfed by phagocytes, in this case, epithelial cells, and also by macrophage, and also using an in vivo clearance model and using alveolar macrophage as well. So all this data supporting that breaking apart may potentially play a role in cell clearance. Next, we decided to look at whether they play a role in apoptotic bodies do play a role in intercellular communication, because quite a lot of different groups have shown that apoptotic bodies can carry various biomolecules to facilitate intercellular communication as well. And we decided to use the monocyte system because even though that we were arguing that the formation of this breaking apart facilitates cell clearance, the gut feeling is that this is actually a pretty messy process. They actually do form quite a lot of vesicles, possibly make it harder for phagocytes to actually engulf. So we think that this process may be probably play more important role in the communication between the dying cells and other healthy cells. And we decided to use an influenza model. So basically, it's known for a very long time that when monocytes is infected with influenza virus, it can actually cause them to undergo apoptosis. And our idea is that we're wondering, if that happens, does the fragment of the dying cells contain viral particle, virions, or viral protein or RNA? So what we did was we've got some human monocytic THP1 cells. When we infect them with the influenza virus, which is the PRX strands, they can cause them to undergo apoptosis. But what's important is that the cells can actually form the formation of those beta apoptopodia and undergo fragmentation to generate apoptotic bodies. We then isolate those apoptotic bodies and trying to see whether there's actually virions in there, and we were able to identify virions either associated on the apoptotic bodies or even inside the apoptotic bodies as well. So the next obvious question is that we were able to infect monocytes with influenza virus, induce them under apoptosis, and they were able to isolate those apoptotic bodies using a FACS-based approach. And then we transfer this to recipient cells, epithelial cells, to see whether the apoptotic bodies could actually aid the trafficking of virions and cause the epithelial cells to become infected as measured by the NP nuclear protein. And indeed, when we do give the epithelial cells apoptotic bodies generated under this scenario, a proportion of them seems to become infected. But what's interesting is that when we kind of repeated the same experiment using a mutant virus called M2SR, whereby the virus lacks the M2 ion channel, and the virus cannot bud out following the first infection, the apoptotic bodies isolated using that approach are not as infectious, suggesting that the virus may actually have to bud out first, maybe then reattach to the apoptotic bodies, and that's how they get trafficked to other cells, at least as a proof of concept. In this study, what we also show is that we identify a drug that can actually block infected monocyte to break apart, but without affecting the apoptosis part. And this drug is called haloprotanol, HALO, which is an antipsychotic. Although it was very effective in doing this in vitro, it was pretty modest. When we trying to block this process in vivo, we can only reduce 20% of the cells to break apart. But nevertheless, we went ahead and did a disease model, whereby we infected some mice with the influenza virus, and then on day 2 is when the peak of the peak of the monocyte undergoing apoptosis. We treat the mice with either HALO or with the vehicle control, and then at day 6 we can monitor the pathology in the lung. And mice treated with the drug that could potentially block the disassembly process seems to do a little bit better with less hemorrhaging. But right now, the data is pretty modest because realistically, we can only use a certain concentration in vivo, and it's not blocking it as well. Hence, the effect is not as dramatic. So right now, hopefully we can identify better drugs or better way to actually block this process and answer the question, whether this monocyte breaking apart and trafficking virus, does it play a role in the whole disease process? So in summary, as summarized by, it's pretty simple, by my daughter Isabel when she was 4, is that when dying cells die, they do undergo various morphological changes, which then eventually the cells would then break apart into smaller fragments, and that they can interact with neighboring healthy cells. But the process is way more complicated. We only have identified a few of the regulators. For example, we don't even know what's downstream of pannexin 1. I'm sure there's other positive regulators in this process, as well as how the fragmentation process goes, how does apoptotic bodies interact with phagocyte. We still need to work that out. But what's exciting for us is that from the drug screen, at least, we have identified a number of drugs that can actually enhance cell disassembly, most of them targeting the pannexin 1 channel. We've identified a lot of drugs that can actually block disassembly. And what's important is that a lot of those drugs are actually FDA-approved drugs. So the dream for us is that if we can work out why we need cells to break apart, and also in particular in disease settings, we have potentially have drugs available to target this process in those diseases. So finally, I'd like to thank my group. They've done an amazing job, as well as my growing list of collaborators that help us to do a lot of the work to achieve that, as well as the funding bodies. Thank you.
Thank you so much, Ivan. It was a great talk. Thank you for sharing your knowledge and data. So for everyone, please type your question in the chat, and we will unmute you. So we start with Hernando Portillo. Please unmute yourself and ask your question.
Thank you, Jan. Thank you so much for your talk. It was excellent. We always have this confounding factor of apoptotic vesicles, apoptotic bodies when we study extracellular vesicles. Do you think that the drugs that inhibit apoptosis can be used if they do not interfere with EV release to avoid this confounding factor?
Yes, potentially. I mean, that's the thing, when you collect other vesicles, it's always, a lot of cells that you use, and, in most cell culture, like, 2 to 5% will die, right? So we haven't tried this ourselves. So for example, I mean, if you say use a caspase inhibitor, pan-caspase inhibitor, whether that will, reduce the level of apoptotic bodies in your sample. But I guess the question is, I mean, apoptotic bodies probably not a major issue usually, because I mean, most people will, do the appropriate spinning step to remove them. What is concerning is probably the smaller, exosome-like or smaller EVs that are also released during apoptosis that you may want.
Well, as far as I understood, even though if apoptotic bodies are larger, sometimes you have some pieces of apoptotic bodies that are of the size of extracellular vesicles, and this is why I'm asking if this could be used, to avoid this confounding factor in our studies in vitro. But
no, it could be done. I haven't done it myself, but I mean, even with some simple experiment, say fibroblasts, they probably release EVs, and you have got back knockout. They don't die, maybe, and then that could be a good way to try.
Thank you.
Thank you. So we go to Gibar Nimata. Please unmute yourself and ask the question.
Hello, sir. It was a very nice talk. I would like to ask is how can influenza virus survive in apoptotic bodies? If the virions are there in the apoptotic bodies, how did they survive in the process of communication? Like, as we know, the virus needs the cell host machinery for its survival. So how is it still infective?
Yeah, the role of apoptosis in influenza virus infection is I guess it's pretty complicated, because there is the virus actually have proteins like PB1F2 that can actually cause the cells to undergo apoptosis at later stage. So whether there's a beneficial effect of that is interesting, but at least in our settings, the virus is able to form virions prior to the cells at least, undergoing apoptosis, because, how cells form this protrusion and apoptotic bodies is actually quite rapid effect. So it can happen within, 3 to 4 hours, well, actually within 2 hours, actually. And then by then, the virions are already formed. So, so therefore, it's possible that, the virions form, and then later on, the cells, it will trigger apoptosis, and then the virions would then, hijack the system. That's at least, the hypothesis that we're working on.
Okay, thank you.
Thank you. So Mona Huttonen, please. Unmute yourself.
Hi, thank you very much for your talk. I was wondering, when you, it's basically a technical question, how can you separate influenza viruses, the viral particles, and then the apoptotic bodies in your experiment, so that when you put that sample on the next cell system, you're sure that you're not just infecting with the released virus particles?
Yeah, that's the kind of like the major problem that we have to deal with in that paper, because I mean, for our own concern, whether we're doing the right thing. So the way that we did in that paper is that we use FACS sorting, to isolate. Okay. And basically, the FACS sorting, you can't sort viral particles. It's not possible. But then after the sorting step, obviously there could still be virus spin hanging around. So therefore, we do the spinning step of 3,000, if I remember correctly, around 3,000 G twice. So obviously, viral particle won't be pelleted at those speed. And then we collected them and then perform our assay as well. So that's the best we can try to do that, and at least that is logical to us. But there's always the concern, in every experiment, there could be small amount of virus being carried over and so on. So, but we did our best to eliminate that.
Okay.
I'll follow up to that question and ask you whether there's a type of particle in between clean apoptotic bodies and viruses that are sort of a mixture of the two.
Oh, as in, apoptotic bodies that generated not from influenza virus, and then, so say for example, if you have a apoptotic bodies, if you influenza virus, what will happen? Is that what you address? Yeah. Yeah.
So, I mean, how distinct are these particles from each other? How exactly different are they, if you see what I mean?
Oh, okay. They're definitely very different in size. At least the apoptotic bodies that we isolated from the what do you call it? From the monocytes, they are between visually they're between 1 to 2, 3, 4 microns. Whereas viral particles would be, in the range of, very smaller nanometers. Of course, there are the what do you call it? The one that, filamentous version of it that could be a bit bigger, but they are hugely different in size. So,
yeah, I don't want to hijack the discussion here, but my view of it, maybe it's wrong, and correct me if I'm wrong, is that when you have apoptosis, you have large bodies, right?
Yep. Mm-hmm.
Big blebs, basically. But you also get small vesicles that kind of are the size of an exosome.
Yes, that's right.
So size is perhaps not the only factor.
Yeah, definitely. But we haven't actually. So that's the kind of area that we hopefully will get onto, because our expertise right now is to apoptotic bodies. But we fully acknowledge that it's quite interesting about during apoptosis, how does the other smaller EVs come into play? When do they get released? And whether they actually also play a role in this process, we actually don't know, because we haven't done the experiment to isolate those under those conditions.
Thank you. So we go over to Anata Sundararaman, please.
Hello.
There you are. There you are. Okay.
Thank you. That was an amazing talk. I was just wondering if this, all these molecules, ROC1 and Panaxin, if they are the same pathways that play a role when the tissue has to establish homeostasis and excess cell numbers have to be extruded, and whether this is also true for the process of entosis when the cell has to actually push into another cell. So if there are conserved pathways between the release of apoptotic bodies and those processes.
No, we the quick answer is we don't know. We haven't studied them in like entosis, where a live cells, kind of like, grab onto another live cells and goes in them and still maintain a life. And obviously ROC, plays a role in the pushing extrusion process as well. We don't know in that case.
Thank you.
Sorry.
So we go to Andrew Devitt, which is related to the discussion we had a minute ago. Andrew, please state your question.
Hi, Ivan. Really lovely talk. It's great to hear other people working in the field of cell death and vesicles. We are so often a contaminant in other people's systems, but I agree that I think we've got a lot of good biology here. You've answered the question about what you feel your definition of an apoptotic body is in terms of size. And then Jan has followed up with this question that we have inherently about the heterogeneity we see between small ones coming perhaps from multivesicular bodies and large ones from the plasma membrane. So the next question really for me is, do you see whether your apoptotic bodies, these larger bodies, influence the phagocyte in the way it deals with its meal? There was some very early work from Valerie Fedok that showed that particle digestibility changed the receptors the recipient cell was using for dealing with apoptotic cells. So I'm wondering, do you see more alpha V beta 3 uptake mechanisms coming online and reduced PS recognition, or vice versa?
No, this is a great question. I mean, something that we haven't done is now looking at the other side, right? So, kind of have been spending a lot of time focusing on the apoptosis part, where I come from the same area as you are, the clearance part, what receptor is using. I don't even know, what receptor is actually engulfing. And also, I guess the question that we have always been trying to answer is that, if the phagocyte actually encounter different size vesicles, would they then again change the properties? Now we have the tools to do that. Hopefully we can address that, without not too many confounding factors, because a lot of the things that we play around with, like Pennexin 1, plays other roles in that process. So I'm a bit concerned about that. But that's definitely the case. Whether that will, maybe the larger particles use different receptor, or maybe different opsonin will play a role. And also, once they're engaged, does it change the profile, of the phagocytes?
Yeah, I mean, they're the same questions. We're at the other end of that size spectrum. We've tried to focus on the smaller, perhaps more complicated, well, more difficult to analyze vesicles, but we should chat offline sometime. That was great.
No, definitely.
So Renata Skovornova does not have a functioning mic, so she had a technical question about the isolation of apoptotic bodies. Is it simply centrifugation? And the size, and how does the centrifugation by itself influence the structure of the apoptotic bodies? Do you use a swinging bucket rotor? Do you have a cushion? Do you pellet them? Things like that.
Yep. So we actually use the cushion. So I think separating apoptotic bodies is definitely a way that we need to improve on. So we want to move on to using a column-based approach. But the tricky thing is that we can't spin them for too long, because the concern is that they will actually undergo lysis. We have to be very careful with them, because they are definitely more fragile compared to other vesicles. A lot of the assay that I've mentioned before is done by FACS approach. So therefore, once you isolate them and we can visually look at them, they seem to have similar morphology as we would when we do time-lapse microscopy with them. So therefore, we're not squashing them or fusing them in that way. But we definitely need to improve on other methods of isolating them, because the differential centrifugation is effective to some extent, but it's not the best for us.
Thank you. So we shift to Sharif Ibrahim. Please unmute yourself.
Hello. Thank you for this great talk. I was wondering if as I understood, the apoptopodia and beaded apoptopodia are two different pathways. They are two different processes, or if they are just two stages of the same process. And if they are two separate processes, is this information? If there is information that if they do different things, or the final product is different and they give different types of apoptotic bodies, or?
Yep. So we kind of approach, well, I mean, we're still working on that, But what's interesting is that we think that the ability of the dying cells to form this protrusion are regulated by the same process. But whether they become beaded or not, it depends on another step. So as I mentioned to you before, the cells undergo, say for example, I use the example of, at least in the in vitro system, the surrogate T cells. The cells form really big blebs. And then in that scenario, when you give them the pannexin inhibitor, they form protrusion. They're really thin, call them apoptopodia. But what's interesting is that at the same experiment, if we block the step 1, which is the blebbing step, so the cells will die. They don't bleb, but they form the protrusion. A lot of the protrusion will become beaded. So therefore, we feel that whether they become beaded, it depends on another step prior to that, and that's blebbing. In terms of the content, we did a very simple analysis. We're asking the question, do they contain nuclear proteins? Because traditionally, people argue that nuclear protein fragmented go into apoptotic bodies. We find that if the cells undergo really big bleb first, and then from the protrusion, nuclear material can get in there. But when you stop the blebbing process, the still will form the beaded protrusion, forming really small apoptotic bodies generated from the beaded apoptopodia. They don't contain nuclear protein. And we are right now, that's those 2 scenarios which have collected the vesicles and done the proteome analysis, but I haven't seen the data myself, but it's there. Yeah, but they are different. Yeah.
Thank you very much. Claire Hill.
Hello, yes, thank you very much for your talk. I'm just interested in the non-apoptotic functions of caspases, and I'm wondering if the factors that you mentioned, such as pannexin 1, have an influence in EV release in healthy cells, and if caspases have a role even in this non-apoptotic scenario.
Pannexin can be active, for example, if I use pannexin as an example. So pannexin can be activated via quite a lot of different mechanisms. A number of different caspases could cleave it, but then it can also be activated by mechanosensing and so on. What we haven't done though, I think something that me and Suresh Methavinan has always chatted about doing, but never got to do, is, for example, now obviously we have got the tools. We've got the pannexin knockout or drug. Do they influence the release of other EVs, like what you mentioned in healthy cells? We haven't tried. So basically, we want to compare, are this machinery plays a role in other EV release in healthy cells, or even oncosome or whatever? We haven't tried, but something that we hopefully will get into.
Thank you.
So we have another question from Andrew. David?
Yeah, sorry, Jan, I'm dominating the conversation now.
That's fine. I mean, so do I.
Your zebrafish stuff, however early that work is, looked great. That intravital microscopy to look live at vesicles being produced is super. But the annexin 5 construct you were using means you're looking at those vesicles that are PS exposing. And I wonder if you had a feel for whether all of your ApoBDs are PS exposing or a subpopulation?
At least in zebrafish setting, we don't know. Unfortunately, the problem with the annexin 5 is it will actually bind to necrotic cells, and necrotic vesicles will have the same thing. So we actually did see the cells undergoing necrosis and releasing all this in the necrotic vesicles. In vitro studies, we have got a lot of vesicles that generated apoptosis. We look at under the microscope. We observe them breaking apart. Some of them are really annexin 5 low. So therefore, we are definitely missing a lot of them based on annexin 5. Whether some of them will eventually become PS positive because of lack of energy, or eventually enough caspase will affect the flipping process, I'm not too sure. But from our point of view, if we do things in vitro, we definitely get a population of annexin 5 negative vesicles that I'm convinced they're apoptotic bodies, but they're annexin 5 negative.
Okay. And then the next step on from that is, do you see different functions from your ApoBDs, beaded or non-beaded? Do you think they might behave in a different way?
Not sure, but usually when they're beaded, they're usually smaller. When they are, so again, whether the size plays a role. But one thing that is very clear to us is that the contents of those are definitely different. And hopefully, you know, we've just recently done a proteome analysis again with Suresh, and then hopefully we can actually, you know, provide that data for everyone to see. Yeah.
So I guess just to elaborate on the point, I'm just wondering whether the beaded vesicles are almost tethered to the cell so that they are being encouraged to act locally to the dying cell, and the non-beaded are released for a more systemic effect. I guess that's the idea I was trying to seed with you, that we might talk about.
Yeah, definitely. The local and distal thing is a very interesting topic that we are working on right now, and it's pretty cool.
Cool. Thank you.
I'm going to throw out a little bit of a difficult question, maybe, just for a general discussion. That is, I presume apoptosis is part of health and part of our normal regeneration of cells in the body, and probably contributes to vesicles present in interstitial tissues and so on. So how many apoptotic bodies do we have in our body, in our whole human body at one time, and does it change over life, and is it important for maintaining health?
Very good question. I think we have started the approach of looking at endothelial cells simply because if endothelial cells do die, we can easily access, you know, the blood and so on to look at them. And we are trying to track that process in different settings. You know, do we, you know, say for example, we have got a particular condition, we isolate the blood. Can we track how many endothelial apoptotic bodies are there in there? It's still at early stage because, but we do, you know, we can easily identify, you know, a lot of apoptotic bodies, even in, you know, say, I guess in the animal system, the healthy mice, we can see a lot of, you know, apoptotic endothelial cell bodies, and whether that would change in under disease setting is what we're trying to elucidate.
Thank you so much. That concludes the discussion. Thank you everyone for participating. I give the word to Carolina, and thank you very much, Ivan, for a great presentation and discussion.
Thank you very much, Ivan. It's a very great talk. I think we have a lot of things to learn today. We learned about the regulators or apoptosis. We learned about how virus-induced apoptosis is happening. And then also the relationship between membrane blaving and cell protrusion, and also an isolation of the apoptotic bodies and more. I think that's a great sort of summaries on all that you've done, and I'm sure there's much more that you can actually tell us but we have to cut it into 30 minutes. But it's very great talk, and thank you so much for sharing it with us.